usurping lipids for energy metabolism. As I said before, the pathogenesis of DHF is characterized by vascular endothelial cell leakage without cell damage. And this is thought to predominantly be mediated by cytokines. Now, I've highlighted direct entry as one mechanism of cytokine production, and this is what I'll be talking about throughout the talk. However, the overall pathogenesis of dengue is thought to be a little more complicated. For example, during secondary infection, there are dengue virus-specific antibodies to a primary infection that are non-neutralizing during a secondary infection. This isn't necessarily all antibodies, but a subset of these antibodies can bind but not neutralize the virus, and in fact enhance uptake of the virus into Fc gamma receptor containing cells. This is the antibody-dependent enhancement of infection. This leads to increased viral load and increased cytokine production. In addition, again, during secondary infection, there are dengue virus-specific T lymphocytes that react poorly during secondary infection. Instead of neutralizing the infection and being protective, they can actually enhance cytokine production. Other less common mechanisms are NS1-mediated complement initiation, which enhances cytokine production. And anti-NS1 antibodies can also mediate enhancement of cytokine production from the vascular endothelium. One last mechanism is called antibody-dependent cellular cytotoxicity, or ADCC. This is where binding of antibodies on the cell surface target those cells for cellular cytotoxicity mediated by natural killer cells. It's unclear whether this mechanism, which has been seen in the lab, if it's protective or pathogenic in the context of infection. And in fact, the current data is that early activation of NK cells is actually protective. However, overall activation can again enhance cytokine production. This all leads to an enhancement of this plasma leakage phenomenon, again, without vascular endothelial cell damage. Cytokines closely associated with DHF and vascular permeability in particular include tumor necrosis factor, or TNF, and interleukin-8, IL-8, which I'll be covering today. Dengue virus induces IL-8 from a wide variety of cell types. Epithelial cells, endothelial cells, liver cells, dendritic cells, macrophages, and monocytes are included in this. And IL-8 is classically known as a neutrophil chemoattractant. However, it will attract any leukocytes that contain the CXCR1 and CXCR2 and or CXCR2 receptor. In addition, it acts directly on the vascular endothelium, upregulating the expression of adhesion receptors for recruitment of cells. And then in regards to vascular permeability, IL-8 induces the formation of gap junctions to allow the passage of cells through the vascular endothelium to tissue, the site of infection. IL-8 secretion is highly regulated. This is a map of the pathway. IL-8 can be induced by TNF. It will also induce itself, and it can be induced by viral and bacterial infection. This induces a signaling cascade and ultimately activation of transcription factors. Transcription factors translocate to the nucleus, where the IL-8 promoter is activated, and IL-8 mRNA is produced. However, at this point, there's an additional level of regulation, where IL-8 mRNA is usually degraded unless signaling activates mRNA stabilization. At that point, in most cells, mRNA is translated and immediately secreted. In addition, IL-8 actually has proviral properties in that it has been shown in the context of hepatitis C to interfere with the antiviral actions of interferon. Therefore, it is advantageous for the virus to induce IL-8. severity of infection, and that was previously shown. Negative controls had a basal level of IL-8. This is patient data. Dengue fever had some IL-8, did have enough regulation of IL-8, and in various grades of dengue hemorrhagic fever, that was actually increased. However, the exact role and reason that IL-8 is induced during dengue virus isn't clear. Previous work from our lab, my predecessor, showed that dengue virus NS5 was the only viral protein to induce IL-8. She cloned each of the 10 viral proteins, expressed these in HEC293A cells, and measured IL-8 secretion, and saw only that dengue virus NS5 was inducing IL-8. So at the beginning of this work, we knew that NS5 induced IL-8 secretion. In addition, we knew that there were multiple dengue viral proteins interacting with very important signaling pathways. The IL-8 promoter is regulated by AP1, CBP, and NF-kappa B, and additionally negatively regulated by OCT1 and NF-kappa B repressing factor, or NRF. And Carey had seen that NS5 upregulates CBP and AP1 to a lower level, NF-kappa B. These three transcription factors are also very important to the activation of RANTES, TNF, IL-6, and IL-1 beta. These are other cytokines that are also associated with dengue virus. Now, a little more complex is the fact that Carey had shown when she used pharmacological inhibitors that she could have IL-8 secretion with decreased IL-8 promoter activation, increased IL-8 secretion with increased IL-8 promoter activation. And this actually led me to conclude that dengue virus IL-8 promoter activation and secretion are two separate but related processes. Therefore, for this work, I focused much of my time on transcriptional activation. I'm going to present this in 3 parts, covering multiple dengue viral proteins contributing to the transcriptional activation of IL-8 during dengue viral infection. I'm going to also show that expression of dengue virus NS5 activates mRNA of other cytokines regulated by the transcription factors, AP1, CBP, and NF-kappa B, which are also important to IL-8. Dengue viral proteins that activate the IL-8 promoter do so by activating one or more of the individual transcription factors, AP1, CBP, and NF-kappa B. And this is presented across 2 aims. Aim 1, looking at the activation of the IL-8 promoter by infection and with proteins, and aim 2, characterizing the activation of the transcription factors. To look at transcriptional activation, I use the luciferase assay, my favorite assay of all time. To do this, we plate HEC293A cells, and 24 hours later, we transfect them with 2 plasmids. One has the IL-8 promoter driving firefly luciferase expression. The other uses the constitutive herpes simplex virus thymidine kinase promoter to drive Rhinella luciferase. 24 hours recovery, and then we infect with dengue virus. At varying time points after infection, we lyse these cells, and we take these lysates and put them on 2 plates. Add cholintirazine to 1 plate. This is the substrate for luciferase, for Rhinella luciferase, and produce a basal light signal. And this is our transfection control. We add luciferin to the other, which is the substrate for firefly luciferase. In our pcDNA negative control, we always are negative. We use MAL and a TLR adapter protein as our positive control, which is always positive. And then we use increasing MOIs to show dose-dependent induction of the IL-8 promoter. This data is shown here. This is set to fold induction over pcDNA, the negative control. In addition, we use C636 mosquito cell supernatant as a negative control for the virus infection itself. And we use dengue virus increasing MOIs, 0.1, 1, and 2.5. And we measured at 24 and 48 and 72 hours. And we didn't see activation of the ILA promoter until 72 hours post-infection. This focuses on the ILA promoter activation, but we can correlate this as well with ILA mRNA production using our RT-PCR. We use the TACMAN system. In this case, we have primers and a probe specific to the ILA mRNA. The probe contains a quencher on one end, a fluor on the other, and when these are attached to the probe, there's no light produced. As polymerization occurs, the quencher and the fluor are released into the supernatant, and with increasing cycles of polymerase chain reaction, we end up with increasing light signal. And so here I've shown the maladaptor protein as the positive control. Again, this is set as fold induction over pcDNA, the negative control.